Hello students, I am Smriti Pillai, lecturer, Biotechnology and Microbiology department. So the topic which we will be covering today is oxidative phosphorylation. This oxidative phosphorylation process is an energy yielding metabolism that takes place in the aerobic organism. It involves the reduction of oxygen to H2O with the electrons being donated by NADH and FADH2. So what are these NADH and FADH2? These are the electron carriers. So this process occurs in mitochondria. So where does this NADH and FADH2 comes from? So this NADH and FADH2 comes from the process called as cellular respiration. This is an glucose oxidation process which involves glycolysis, pyruvate decarboxylation and the citric acid cycle. So starting with the glucose oxidation, it undergoes a glycolysis step where one NAD is converted to NADH and that gives rise to pyruvate. This pyruvate then undergoes pyruvate decarboxylation to form acetyl-CoA. Here also NAD is converted to NADH. The acetyl-CoA then enters the citric acid cycle or the Krebs cycle where NAD is again converted to NADH and FADH is converted to FADH2. So from this NADH and FADH are the electron carriers, and all the electrons that are being released from the conversion process, they are all being stored in the form of NADH and FADH2. So these NADH and FADH2, these are being taken inside the matrix where these are being converted into NAD and FADH, and this conversion of NADH and FADH into NAD and FADH is by the release of the electrons, and these electrons are further being used to pump the protons out of the matrix. So the whole process of oxidative phosphorylation takes place in mitochondria, where the major aim is the reduction of oxygen to H2O with the electrons being donated by NADH and FADH2. So the mitochondrial respiratory chain, it consists of series of sequentially acting electron carriers, most of which are integral proteins with prosthetic groups capable of accepting and donating either one or two electrons. So starting with the electron carrying molecule, that is NADH. So here there are three complexes being shown. Complex 1, that's called NADH coenzyme Q reductase. Complex 2, that is called coenzyme QH2 cytochrome C reductase, and the complex number 4, that's called cytochrome C oxidase. In this, in the first complex, the NADH is being reoxidized to NAD plus and H. Here two electrons are being released, which will be received by the FMN and FES, which are the prosthetic group present in complex number 1. Then the two electrons will be then accepted by the coenzyme Q ubiquinone. This ubiquinone will be accepting one electron to be converted into semiquinone, and then again one more electron will be accepted by the semiquinone to be converted into ubiquinol. So one electron is accepted by ubiquinone to form semiquinone, and then again an electron is accepted to form ubiquinol, that is QH2. Further, these electrons, when there will be a transfer of electrons from complex 1 to QH2, that is ubiquinol, there will be a pumping of 4 protons from the matrix to the intramembrane space, because as the electrons are being transferred from complex 1 to complex number 3, these transfer of electrons, there will be large amount of energy that will be stored in the intramembrane space, and these energy that will be stored will create an electrochemical gradient across the membrane. So the transfer of electrons from complex 1 to complex number 3 will give rise to a large amount of energy that will be stored in the intramembrane space. Now further, the electrons that are being accepted by the FES in the complex number 3 will be accepted by the cytochrome. Here the cytochrome is of 3 types: cytochrome A, B and C. Cytochrome A is again of 2 types, A and AA3. B is of 2 types, BL and BH, and the C of C1. So here when the electrons will be transferred onto cytochrome C, here also 4 protons will be pumped from the inner matrix to the intramembrane space. Then further, when the electrons are being transferred onto the fourth complex, that's called cytochrome C reductase, it will be accepted by the copper and there will be cytochrome A1, copper B to cytochrome AA3. Here, while transferring on, there will be again a pumping of 2 protons from the matrix to the intermembrane space, and the final electron acceptor will be oxygen. All the electrons that will be transferred from the reoxidation of NADH will be accepted to the oxygen, and this oxygen will be combining with the H plus ions present in the matrix to give one molecule of H2O. So here an oxygen is being reduced to give H2O. So the first complex, when there will be transfer of electron, that will be pumping on 4 protons, then it will be transferred on to ubiquinone, then to complex number 3, that will be pumping on 4 protons, then to cytochrome C will be carrying it on to the complex number 4, where it will be pumping on 2 protons, and the final electron acceptor will be oxygen combining with the 2 protons in the matrix will be giving one molecule of H2O, and the enzyme that will be catalyzing the NADH to NAD plus process is the dehydrogenase enzyme. So the total protons that will be collected in the intramembrane space will be 4 plus 4 and 2, that will be 10 protons. So when there will be an reoxidation of one NADH molecule, there will be total collection of 10 protons in the intramembrane space. Now this is for the FADH2 electrons. The complexes that are involved in this are complex number 2, complex number 3 and complex number 4. Complex number 2, this is also called succinate coenzyme Q reductase complex. Here there is a conversion of succinate to fumarate, and the reaction is catalyzed by succinate dehydrogenase enzyme. This succinate dehydrogenase enzyme is an integral part of complex number 2, and this process takes place in citric acid cycle. So the conversion of succinate to fumarate gives rise to 2 electrons that will be accepted by FAD. FAD will be converted to FADH2, and then further reoxidation of FADH2 will be giving out 2 electrons that will be accepted by the FES. The FES will be transferring the electrons to ubiquinone, and this ubiquinone will be again accepting an electron, will be accepting 2 electrons to form ubiquinol. These electrons will be further been transferred on to complex number 3, and the transferring of electron from the complex number 2 to complex number 3 will not lead to any pumping of protons from the matrix to the intramembrane space. Then the electrons from the complex number 2 will be transferred to complex number 3. Here the electrons are accepted by the FES, then further electrons are transferred to cytochrome C1. Here when the electrons are being transferred in complex number 3, there is pumping of 4 protons from the matrix to the intramembrane space, and then the electrons are being accepted by cytochrome C, that is a mobile carrier. These electrons will be then carried on from complex number 3 to complex number 4. In complex number 4, the electrons are being accepted by copper A, then cytochrome A1, copper B and cytochrome AA3. Here where the electrons are being transferred, 2 protons will be pumped from the matrix to the intramembrane space, and the final electron accepted in complex number 4 is oxygen. This oxygen combines with 2 protons from the matrix to give one molecule of H2O. So here the conversion of succinate to fumarate, that will give electrons, and then further reoxidation of FADH2 in the complex number 2 will give electrons that will be transferred from 2, 3 and 4, and the final electron acceptor being oxygen, that will be then reduced to give H2O. This whole process will be pumping out 6 protons to the intramembrane space, and these protons that are collected in the intramembrane space, that will pass again through the 5th complex, that is also called as the ATP synthase complex or the 5th complex or F0F1 complex. Here for every transfer of 4 protons from the intramembrane space to matrix, there will be synthesis of 1 ATP. As 4 protons, they are being transferred from intramembrane space to matrix, these 4 protons, 1 ATP will be synthesized from ADP and PI in the matrix portion. So the difference from FADH electrons and NADH electron is in NADH electron there is involvement of complex 1, 3 and 4 with the pumping of 10 protons,
And for FADH electrons, the complexes that are involved are complex 2, 3 and 4, and the protons that are collected in the intermembrane space are 6 protons. So, one FADH2 that pumps 6 protons in the intermembrane space, and one NADH that pumps 6 protons in the intermembrane space. So, one ATP molecule from the influx of 4 protons, that is one NADH that pumps 10 protons. So, one NADH gives 2.5 ATP, and one FADH2 that pumps 6 protons, that is one FADH2 gives 1.5 ATP. If you like the video, please like, share and subscribe. Thank you.